Hi everyone, my name is Jiawei Ma, and I'm a PhD student of QOP in RMIT University. And today I'm going to talk about the general steps in computational models of fluid particle in respiratory airway. And here are three main topics of my presentation: the mesh generation, CFD simulation, and particle tracking. So apart from the Hashan's advanced meshing methods, our meshing generation is only use the unstructured polyhedral mesh. And for the CFD simulation, I'm going to talk. We're going to discuss the boundary condition and the CFD simulation setup. And for the particle tracking, we are going to discuss the particle release and the particle deposition result analysis. So here's the first part, the CFD meshing. As shown in this graph, on the left-hand side is the STL model, which is generated from the CT scanning data. And on the right-hand side is the polyhedral mesh of the CFD mesh. As shown on the graph, these models has some differences, and for the STL model, this model only contains the triangular elements, and this is only the surface mesh. So after the CFD mesh, we're going to generate not only the surface mesh but also the volume mesh, and this mesh includes all the computational domain. So here's some details of the computational domain. So normally we build the respiratory airway all the way from the nostril to the tracheobronchial region, but in some specific case, we are only focused on the nasal cavity region. So in this model, we only have the nasal cavity and the breathing zone part. As shown in this model, we have built a little box in front of the mannequin's face. This is known as the breathing zone. So during the simulation, the air is coming from the boundary of this breathing zone, and the boundary is set up as the atmospheric pressure, which is the surrounding region. And the air is inhaling from the surrounding region. The air can freely come in and go out from the boundary of this box. And as the other part is the extension. We built this artificial tube in order to replace the lower part of the respiratory tract. And at this case, we apply the velocity boundary condition as the outlet of this artificial pharynx, because in this way we are able to generate a comparably realistic velocity profile at the end of the nasal pharynx region. And so normally we apply the steady state in the simulation, and so we convert the volume flow rate to the average velocity profile at this stage. And here are some governing equations of the CFD modeling, the CFD simulation. So as we know, the CFD is to discretize the computational domain into very small elements, and the computer will help us to calculate the physical mechanism in each element based on these equations. So these equations are embedded into the CFD softwares. So in this case, we use the Fluent, and all of these equations are applied. Here are some examples of the CFD simulation results. The first one is the streamline, which indicates the airflow pattern. And as shown in this graph, the air is inhaled from the breathing zone all the way through the nasal cavity region to the pharynx and lower respiratory tract. Apart from this simple structure, we're able to simulate the heat and mass transfer as well. Here's the mass transfer, which is the humidify function of our nasal cavity. As shown in this graph, we select 3 typical cross-sectional regions. A is the nasal valve, B is the middle part of the nasal cavity, and C is the choana. As shown in each cases, as the air travel at the choana region, all the air has already been humidified, has been almost fully dehumidified. And based on this simulation result, we're able to apply the particle simulation. So in this part, the first stage of the particle tracking is to release the particle. In the NSAS Fluent, we're able to customize the particle release functions. So in the software, we're able to define the coordinates, the velocity, the size, and the temperature of the released particle. So for the coordinate location, we release the particle in front of the mannequin's face, just very close to the nose. And during the simulation, all of the particle will be inhaled into the nasal cavity. And this particle size, we choose from the 1 nanometer to 10 micrometer. It is because in the deposition curve, deposition efficiency curve, there is the U shape. And when the particle size is lower than 1 nanometer, all the particle will be deposited on the nasal cavity surface. And if the particle size is larger than 10 micrometer, the same thing also happened. So we choose the particle size from 1 nanometer to 10 micrometers. And here are some example of the particle deposition result analysis. As shown in this graph, this nasal cavity has been mainly separated into four segmentations. As shown in the top of the nasal cavity, the red region indicates the olfactory region, which is very important, which has very important function during the inhalation. So if we calculate the deposition efficiency all along with the particle size, we can plot this efficiency curve for different subjects, and we can analyze the particle deposition results differs from the morphological difference. And if we add the efficiency, if we add all the regional deposition result up, we can find out the total deposition of the nasal cavity. And this curve can help us to do the validation and compare with the previous studies, and including the simulation and the experimental result. And this is the other way to help us to understand the particle deposition results. On the left-hand side is the discretized particle deposition location on the tracheobronchial region, and we can plot this result as the continued contour on the right-hand side, and we can with this curve, with this graph, we are able to find out which part will suffer a lot of the toxic particles. Also, it will help the pharmaceutical studies to find out which region can be applied more medical dose. And that's all.